Welcome to our video on the power of CRISPR, a revolutionary technology that is changing the face of genetics. In this video, we'll explore how CRISPR works, its potential applications in medicine and agriculture, and the ethical implications of gene editing. First, let's talk about what CRISPR is. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, a mouthful of a name that describes a series of genetic sequences found in bacteria. Scientists have discovered that these sequences allow bacteria to defend themselves against viruses by recognizing and destroying their DNA. CRISPR has been adapted for use in editing the genetic code of other organisms, including humans. CRISPR works by using a guide RNA molecule to target a specific sequence of DNA, then using an enzyme called Cas9 to cut the DNA at that location. This can be used to delete, add, or replace specific sections of DNA. The guide RNA is designed to match a specific sequence of DNA, making CRISPR a very precise tool for genetic engineering. One of the most promising applications of CRISPR is in the field of medicine. CRISPR could be used to treat genetic diseases like cystic fibrosis or sickle cell anemia by correcting the genetic mutations that cause these conditions. It could also be used to create personalized cancer treatments that target specific genes in individual patients' tumors. In fact, early clinical trials of CRISPR-based cancer therapies have shown promising results. CRISPR could also be used to edit genes in human embryos. This is still an experimental technology, but it could eventually allow scientists to prevent genetic diseases from being passed down to future generations. However, this raises important ethical questions, such as whether it is acceptable to edit genes in human embryos, and who gets to decide which traits are desirable and which are not. In addition to its potential in medicine, CRISPR also has applications in agriculture. CRISPR could be used to create crops that are more resistant to pests, drought, and disease, leading to higher yields and more sustainable agriculture. It could also be used to create crops with improved nutritional content, such as rice that contains more vitamin A to combat blindness in developing countries. However, there are also concerns about the unintended consequences of gene editing. For example, off-target effects could cause unintended changes to the genome, and there are concerns about the safety of CRISPR-based therapies. Additionally, there are concerns about the use of CRISPR for non-medical purposes, such as creating designer babies or genetically modifying crops in ways that could be harmful to the environment. In conclusion, CRISPR is a powerful tool that has the potential to transform medicine and agriculture. However, its use also raises important ethical questions that must be carefully considered. With responsible use and careful consideration of the implications, CRISPR could bring about a new era of personalized medicine and sustainable agriculture. But we must be mindful of the potential risks and unintended consequences of gene editing, and work to ensure that the benefits of this technology are shared equitably and responsibly. Thanks for watching. Please like and subscribe.